So thanks, Kathy and Tom, for the chance to come and provide an update on the work that we're doing. Essentially, our project's goal is to define whether or not a biological process termed the glymphatic pathway that's been characterized in rodents is functional and taking part in human biology, and whether it's relevant to the development of Alzheimer's disease. So I'll just kick off with a quick, I think some of the people in the room may have no idea what that word even is, because it's actually a made-up word. But the glymphatic system is, as it's been defined, it's a brain-wide network of perivascular spaces by which the fluid that surrounds the brain, the cerebrospinal fluid, exchanges with the fluid in between the brain cells, the interstitial space. And the movement of fluid along these perivascular channels and along these perivascular pathways is thought to facilitate the efficient clearance of interstitial metabolites and waste, like amyloid beta and tau, which you've heard a little bit about in the last couple of talks, and other metabolites like lactate. This is a new idea in the sense that it sort of flies in the face of conventional thinking about the CSF circulation. The conventional thinking is that the CSF is sort of a passive fluid that sits on the outside of the brain, and it sort of floats the brain and serves a sort of support role. But our data from over the last couple of years suggests that it's much more active and much more organized than that. So a quick kind of, let's see here. Quick view of what that looks like is the fluid, if you inject a tracer into the fluid that surrounds the brain, this is a mouse brain, it doesn't just float over the surface of the brain, but rather it follows very specific anatomical pathways. You can see that it tracks along the surface of the cerebral surface vasculature, specifically arteries that run along the brain surface. You can see that using in vivo multiphoton microscopy, that the contrast moves initially along the pial surface, along these pial arteries, and then as those arteries dive down into the tissue, the CSF follows those vessels and begins to exchange through that space. So then this glymphatic system is this brain-wide network facilitating this exchange in this wash-through. One of the more interesting things that we observed along the way was that this is a biological process that is primarily a feature of the sleeping as opposed to the waking brain. So this is an experiment in which that same process of CSF recirculation through the brain is imaged in animals that are waking and animals that are anesthetized. So this movie is actually rolling. So this is an animal that's in a waking state on the multiphoton scope. And then a short period of time later, it's anesthetized, and then a second injection of CSF tracer is injected, and you see a completely different situation. So what these data demonstrated was that the brain is actually moving in between two different states. One in which this biology is engaged and is rapidly clearing away metabolites like amyloid beta out of the brain, and one in which it's largely quiescent. And that's the sleeping and the awake state. So these same data were recapitulated using natural sleep and awake, not just anesthesia. So the question that's begged from these earlier experiments is whether this process, number one, is present and functional in the human brain. Is this mouse biology or is this human biology? And then the second question is whether it has a role in the development of diseases like Alzheimer's disease, right? So in animal models, we see that this process is impaired in the aging brain. We see that it's impaired in the post-ischemic brain. We see that it's impaired after subarachnoid hemorrhage. We see that it's impaired after traumatic brain injury, suggesting that a failure of this very basic homeostatic mechanism of the brain may underlie these many different disease states. We don't know if that's true. We don't know if that's true in the human brain. So, and similarly, we don't know if that failing process underlies the development of Alzheimer's disease. So there's two ways in which we can go about this. The one way, which is actually the main focus of this project, is to try to do the same kinds of imaging we've done in rodents and humans. Now, we can't stick a human's head under a multiphoton microscope, or at least, I don't know, maybe you could, but it doesn't seem very advisable. But you can approach the same sort of imaging using MRI, and that's what Bill's going to talk about in the second half of the talk, is the attempt to develop clinically relevant imaging modalities to capture this biology. A tack that my group, which is more of a molecular cellular group, is taking is to look for evidence of the involvement of this biology in human tissues, in human samples, and in human data sets that will tell us whether or not there's at least corollary or parallel evidence for its involvement in Alzheimer's disease. So to get at that question, you guys have seen this, or some of this already before. We can look at just the architecture of Alzheimer's disease and how it works. So Alzheimer's disease is characterized by the deposition of extracellular plaques made up of amyloid beta and intracellular neurofibrillary tangles made up of hyperphosphorylated tau. What's interesting about the disease, one you've heard about before, is how it's really intrinsically tied to the aging process. But the second is how it exhibits really classical neuroanatomical pathways of spread. In the case of amyloid deposition, there's very specific regions of the brain that develop amyloid deposition very early, and that deposition then progresses to encompass many, many more widely spread regions of the brain. Similarly, neurofibrillary pathology begins in very specific neuroanatomical locations and then appears to spread perhaps in a prion-like form to involve other regions of the brain. So one of the questions that you can ask is whether regions that are prone to protein misaggregation exhibit a different architecture of the glymphatic system. And so you can look at these different brain regions and classify the ones that represent the Thal early stages or the BRAC early stages as being protein aggregate susceptible versus those that occur later as perhaps being more resistant. And so this is sort of a methodology that was totally copied from a recent paper that came out in Science Advances. But you can ask the question of whether differences in the architecture of the glymphatic system underlie some of this neuroanatomical vulnerability to protein misaggregation. So when we think about the cellular molecular architecture of the glymphatic system, the thing that we focus on most specifically are astrocytes, and specifically the perivascular end feet that ensheath the cerebrovasculature. So about 50% of the surface area of these perivascular end feet is occupied by this water channel called aquaporin-4, and it's anchored there by a series of adaptor proteins, so alpha-syntrophin, dystrobrevin, that attach it to the macromolecular dystrophin-associated complex with dystrophin, which then anchors to the basal lamina at the blood vessel through dystroglycan. So if we look at the expression of all 5 of these candidate genes, we can look at their expression pattern throughout different human brain regions. So using the Allen Human Brain Atlas, we can characterize different brain regions as being aggregate prone versus aggregate resistance. And we can look at the expression profiles of these to determine whether there's a biasing in the expression patterns. And one of the interesting things that we see, if we just take two examples here, aquaporin-4, which is the water channel that organizes water movement through the brain, and dystrobrevin, which is an adaptor protein that holds it at this perivascular localization, you see that regions that are vulnerable to early amyloid deposition, so they're involved in Thal stage 1, they express both lower aquaporin-4 and lower levels of dystrobrevin, whereas regions which are involved later in the disease course or do not appear to involve amyloid aggregation, they appear to have higher levels. So that suggests that at least different brain regions that are vulnerable to different levels of amyloid deposition have a different molecular expression of these proteins. You can see exactly the same sort of thing if you classify things based on whether they're vulnerable to neurofibrillary pathology. So regions that are associated with the earliest BRAC stages 1 and 2, versus the later BRAC stages 5 and 6, versus regions that don't appear to exhibit neurofibrillary pathology. You see a really marked difference in just the simple mRNA expression pattern of the members of this larger macromolecular complex. So work in our group is going toward expanding on this gene list and trying to figure out if astroglial gene networks more generally are one of the factors that underlie regional vulnerability to protein misaggregation. We can also look in histopathological samples. So we have an NIA-funded Alzheimer's Center at Oregon Health Science University that has a huge brain bank that's really well characterized. And so we did a study looking at aquaporin-4 expression and localization across age and in people with Alzheimer's and without. And one of the things that we see is that aquaporin-4, which organizes water movement through the glymphatic pathway, it's really markedly, there are marked differences in its expression and localization when we look at the young brain across, this is the gyrus of the human frontal cortex, versus even the cognitively intact aged brain. You see very, really striking macroscopic changes in the expression pattern of this protein. If we measure how localized it is to the perivascular end feet, we end up seeing that that perivascular localization very strongly predicts Alzheimer's disease status as well as the severity of amyloid plaque density and neurofibrillary pathology, suggesting that, at least within this cohort, that changes in perivascular aquaporin-4 localization are at least associated with Alzheimer's pathology and the development of Alzheimer's disease. It doesn't demonstrate causality, but it's consistent with the idea that this biology is involved in this disease process. The last one comes, the last little vignette is taking a very different view of aquaporin biology in human disease. So this is genetic GWAS data coming from our longitudinal aging studies. So this comes from about 650 subjects who entered a longitudinal aging study around the age of 65, all cognitively intact. Many of them convert to Alzheimer's disease or MCI. Many of them don't. But if we look at naturally occurring single nucleotide polymorphisms in the human aquaporin-4 gene, we see that of the 5 that we queried, all 5 are associated with either more rapid neurocognitive decline, in the cases of these SNPs here, or slower neurocognitive decline in the case of these SNPs here. So these are clinical measures of cognitive function and also some other measures of executive function and memory. So what this suggests is that we have data along multiple parallel lines that suggest that variation in the human population, in the genetic structure of at least this gene aquaporin-4, which is a major piece of the glymphatic pathway, in the histopathological studies, and even in other data sets in cognitively normal subjects, suggests that there does seem to be an association between what we know now to be pieces of this biology and what we see in human disease. Now, this is the worst kind of correlation. This is what we do when we can't see the function of the biology yet, which is what Bill's going to talk about next, is our efforts to actually see what's actually happening.
Well, can you hear me okay? Great. Well, thank you, Kathy. Thank you, Tom, for the invitation to present. And it's been a great project. It's been a lot of fun. It's been a lot of challenges. And as Jeff mentioned, one of the primary outcomes that we'd like to do is to investigate this novel biology in human subjects and do it non-invasively, and also do it in a way that we can map throughout the brain, because there may be dysfunctions that are regionally localized. So to do that, MRI is really in many ways an ideal platform for it. It's non-invasive. It provides excellent spatial resolution and coverage. And there are a large number of biomarkers that we can investigate, including some that are very novel in energy metabolism, which I think is really central to some of the processes that we're looking at. But of course, it's not perfect, right? Anyone that's had an MRI knows that these things are noisy. It's a small space. They're very sensitive to motion. And there are strong fields that are required, strong static magnetic fields, strong radio fields. They're not ionizing, but they are fields that can couple in with some of the other devices that we need to characterize sleep. So one of the challenges, and Jeff and I have been talking about this for a while, since the glymphatic process is a sleep-dependent, how do you get somebody to sleep in one of these instruments? So we've been spending a lot of time kind of re-engineering what we do to make the systems quieter, to make them more comfortable, and then to couple them with devices like EEG, electroencephalograms, which couple very strongly with magnetic fields to be able to stage sleep. So up here, we show just some of the testing that we've gone through. This is an Agar Phantom, so you can make these things. We can 3D print molds from MRI pictures, volumely rendered, and produce these casts, and then Agar pour them so they have the same conductivity of the human head. We can use our brain mask, which has all the electrodes, put it in the MRI environment, and then do very detailed temperature mapping to make sure that we're not doing any harm. It's really important. I think the fields that we deal with are very high. We want to make sure everything we do is safe. So we've gone through a lot of testing with these different phantoms. These are actually really interesting Halloween props. You leave them around a lab for a couple of days, and they grow some very, it's basically an agar plate shaped like a head. But you get nice MR signals from it, and we can do very detailed temperature mapping. So we're very comfortable now with the parameters that we're working with and the changes that we're seeing. So this is really fundamental in being able to stage sleep. Of course, the brain is very expensive to run. It's metabolically very expensive. It's interesting to think that it uses primarily glucose and about 100 grams per day. That's about 1/4 pound. That's a lot of glucose, a lot of energy. And of course, most of the glucose is fully metabolized, but not all of it. Some is lost, and that's interesting because there's a whole, and of course, how the mitochondria ages and all of the oxidative processes and the demands it puts on the brain are very interesting to think about as well. So energetics, very interesting. So the magistrating mechanism, the brain is highly organized, right? Neurons, glia, tightly coupled. It's evolved to help neurons process. So this is Magistrati hypothesis focusing on lactate, monocarboxylic transporters that move lactate between glia. This is capillary here. This is a glia cell. This is a neuron. So to shuttle lactate between these glial cells and neurons, and of course sometimes it's going to be exposed to the extracellular environment. And the extracellular environment is one of the things that's very dynamic in the glymphatic, one of the tenets of the glymphatic hypothesis. So brain energetics, so looking at metabolites in sleep. So there's a long history of looking at things like lactate. And back in 1948, Richter and Dawson reported 35% decrease in rat brain lactate during sleep. Those findings have been replicated, and in fact, in a very detailed and nice paper that was published in 1972. And it just indicates that lactate is important. It's recently replicated by Nadergaard's lab at Rochester in mice. And here we're looking at immobility-defined sleep in the mouse. And you can see that during the light phase, the mice are much less active. So this is kind of inverse. So they're more sleep-like. And that's shown on average here for the light phase versus the dark phase. And what they did was a very clever experiment, microdialysis, looking at brain lactate in these different phases of the day. And what they found was a very significant decrease in lactate during sleep. And with that, so depletion of lactate, but accumulation of lactate in cervical lymph nodes. So it suggested that this extracellular lactate was being shuttled out of the brain more effectively during the sleep phase. And they did a number of different manipulations to convince themselves that it was actually, as Jeff mentioned, this interstitial fluid flow that was responsible for it. They did things like modify the CSF production, knock out AQP4 and 4, as Jeff mentioned, is very important in glymphatic function. So it seems very like a very compelling biomarker in the small animal. Of course, brain is highly organized. There's very small spaces between brain cells. In the mouse cortex, during the sleep state, the extracellular space is enlarged compared to the awake state, and that increases about 60% based on these measurements. And that's the mechanism that provides a lower impedance pathway for washing substances out of the brain. That's the tenet of the glymphatic physiology. So we're able to do that. This is a system we just installed over the summer. It's a new 3T MRI. It's quieter, which is good. It's got a lot of accessories that make it very amenable to doing these kinds of measurements. So we're very interested in metabolism, and here's a proton spectroscopy from this region in the back of the brain, this volume element here. And you can see very nice peaks for N-acetylaspartate, which is a neuronal marker, creatine, which is involved in energy metabolism, choline, methyl protons from choline, and also there's a peak right here with lactate. So we can follow this, do a time series, put somebody into the magnet, let them sleep, and follow that. So we did that with, so these studies are going on now. This is an example from a healthy young control, 32-year-old woman who was sleep deprived. She came in early in the morning, and we did some anatomical imaging and then started the spectroscopy, which is collected every 2 seconds. We block average for 2 minutes, and you can see the lactate signals about 700 micromolar during the awake state, very consistent. When she went to sleep, we see a sharp decrease in the lactate, comes up. She woke up briefly here, went back, and then came to sleep. And all of this is corroborated with physiological measurements that we collect in addition. So it's really interesting to think about this now in terms of as one of the biomarkers that we can use to estimate lymphatic function. And you can think about it in terms of just testing people. We now have the capability to make them comfortable, to grade sleep while they're in the scanner, and then to non-invasively measure how this lactate is changing. And you can imagine that as the brain ages, the structure is very, very different. So the coupling between neurons and glia may change, and the way that it changes across the sleep cycle may be very, very different, and it may provide a very early readout for when this system is going awry. So this is one biomarker. We have a number of others that are a little bit noisier that we've actually done a lot of development work on that we're collecting data with now. And so it's very exciting to investigate these measures and to start to see some of these results. And, but one of the I think the best aspects is bringing together a very diverse team, and very few mechanisms do this. So it's really challenged a lot of the ways that we do our measurements. And some of the people that have, Zelda Yeldiv, who's a postdoc that just came on about 6 months ago, is driving the signal processing part of it. Ian Tagay is doing a lot of the MRI. Corey Wyatt is an MR physicist who's helping us design smarter MRI techniques. So with that, I'll open it up for questions, and thank you for your attention.
Metabolic requirement for lactate, but lactate is also an acid.
Mm-hmm.
Is it possible that the brain pH is changing between sleep and awake cycles? Is there a way to measure that with MRI?
Yeah, so there is a way to measure that. We do that fairly routinely. One of the other things, you can measure the chemical shift of many of these compounds, and they're pH dependent. So that's a measure that we can pull out. We haven't with this data yet, but it's something that we can certainly look at. So I don't know the answer to it, but it's something that we can investigate. Yeah.
What happens to oxygen consumption during sleep, and
Yeah.
can you monitor that with MRI?
Yes. So there are ways to measure brain oxygen utilization. They tend to be. There are ways that you can do it with isotopes. Oxygen 17, of course. We've got a 7T MRI instrument that is well suited for these multinuclear. They're expensive. Low resolution. There are other techniques that are using basically a variant of the BOLD technique, the blood oxygen level dependent, where you can measure the oxygen level in veins or arteries that are feeding different regions, and look at output from veins and look at that differential. There are ways that you can get at that indirectly that I think would be very interesting. We also have a nuclear medicine group that's very skilled at making molecules that are sensitive to different oxygenation states within the brain.
Just a really quick, is there any drugs that open up the glymphatic system? Anything known yet?
So there are some that are modulators of the AQP4 channel, but whether or not that would actually do something in this state, it remains unclear. The evidence so far out of the animal studies suggests that manipulation of noradrenergic tone within the cortex has been the primary way by which things have been modulated in experimental settings. So we're actually doing work with the noradrenergic antagonist prazosin, which is used currently clinically for the treatment of PTSD, because one of the things underlying PTSD is thought to be sort of overactive cortical arousal that extends into sleep, leading to nightmares and things like that. So prazosin is a drug that we're looking at right now as a way to turn things on or turn things off.
Thank you very much.
Thanks.